Okay, once again, good afternoon, and thank you for make yourself available to join this live talk series 2021 on pharmaceutical chemistry. Okay, so as introduced by Dr. Keng Pecina, the title is about, do you want to create a drug to cure disease? Okay, so in this talk, I will share mainly of my real own experience, okay, and as well as a little bit basic introduction about how the drug development or drug discovery process happening. Okay. I'd like to begin the talk this afternoon by sharing about my journey, my own journey. So um, I did my Bachelor of Pharmaceutical Sciences back in 1994. It was a 4 years course at that time, so I completed my first degree within allocated time, 4 years, until 1998 in Hiroshima University, Japan. And in this 4 years pharmacy program, we have to perform a research project, okay, in our final year. So in my final year, the research project that I conducted, okay, was on design and synthesis of pentamidine dimeric zinc cyclone complex as a DNA minor groove binder. So pentamidine here is a name of one particular drug that the mechanism of action is actually targeting DNA sequence. Okay, so in this project, I designed a combination of actual drug pentamidine with one molecule complex that we've been studied in my research lab at that time, which is called cyclone complex. So with this combination, we try to make this combination drug more selective, okay, into as a DNA minor groove binder target, right? So with this great interest, okay, in chemical synthesis, okay, and then the curiosity in try to look for new drug, okay. So I pursue my study to master level in 2000, okay, until 2002, at the same university actually, and so the exciting years, okay, as my research journey began from there, and as one of the outcome, okay, I able to publish one high impact research paper, and had opportunities of presenting my research work at conferences, okay, and that interest continue, okay, until I further my study to PhD in 2002 until 2005. Okay, so in this 3 years period, obviously, I gained more interest in research. Okay, and I also enter a new so-called area, okay, in my research study, which is called supramolecular self-assembly chemistry. And as an outcome, again, I able to publish one high impact research paper, okay, at that time, and then of course opportunity presenting my research work at many international conferences. And once I graduated from PhD 2005 until now, is where I started my career in academia, with continuing conducting research, writing a paper publication, as well as supervising postgraduate students, and also growing my research linkages with many universities and also institution researchers. In this slide, okay, so as a title, why do I choose to be in academic medicinal chemist career? So I like to share this, I will say, a very unique own experience. Okay, when I was in the high school, okay, as you can see on the screen at the left side, there is a picture of a cell. Okay, so you can see that the nucleus there, mitochondria, okay, and a few other organelle. Okay, so I was biology nerd, okay, during my high school. Why I say that? Because biology was subject, the pure science subject that I was really confident. Okay, I always get 99% over 100. Okay, the reason why I didn't get the remaining one mark because of it's a general, I think, that you won't get the full mark for your essay. Okay, so teachers always every time after finish examination, the biology teacher will always announce to the class that if you want to know the correct answer, please ask Zul. Okay, so that's how nerd was I with regard to the biology. And another reason was because I wanted to be a medical doctor at that time. Okay, I think it's really common. Okay, among high school students, if you ask about what you wanted to be, is always like in the medical line. Either you think of becoming a medical doctor or dentist. Okay, and I would say hardly people will mention about wanted to be pharmacist, because I think not many people are really aware about what is the pharmacist job or what pharmacists can do. Okay, so and at that time my chemistry and physics was really average. Okay, as my SPN result, I only obtained credit 3, okay, for my chemistry and physics, while my biology was distinction A1. Then even though I wanted to be a medical doctor, okay, when I went to my pre-university level study, okay, the program was actually a preparation program to study in Japan. Okay, so even though during the interview I told the sponsor that I wanted to do medicine in Japan, and medicine actually is a 6-year program in Japan. Okay, while pharmacy at that time was still 4-year program. Okay, um, so along the way during my pre-U, when I started study a Japanese language, I found that my Japanese language performance was not that good at that time. So I wonder if I still pursue with 6 years taking medicine in Japan, I may not maybe have to repeat year. So may end up 7 or 8 years to graduate. So after think many times, I switched from taking medicine to taking pharmacy, the 4-year program. Uh, even though I honestly I didn't know what actually pharmacy program is all about. Okay, so then as introduced in the previous slide, okay, I went to Hiroshima University, and along the way study pharmacy in that 4-year. There is one module or course called medicinal chemistry. So that is where I learned about this one anti-cancer drug, which is called cisplatin. Okay, you can see the structure of the cisplatin on the screen now at the center. Okay, where the red color PT2 plus. So that is actually showing the platinum metal ion. Why I feel impressed with this cisplatin is because 2 reasons. First is because this is the simplest anti-cancer drug structure that I ever learned. Okay, so it just with one metal ion, platinum, and then you have 2 chlorine atom that link to the platinum. Okay, and then you have 2 amino molecule, okay, group that connect to this platinum. And so that is first reason. Second reason is I learned that this molecule, okay, or this complex, platinum complex, work as an anti-cancer when it is in the cis position. Okay, that is where the name come from. Okay, cisplatin. So, but if it is in the form of transplatin, okay, luckily I prepare a model here. Okay, so if you can see that even though this is not 100% right, just assume that the black black sphere here is a platinum, okay, and this 2 blue is the amino, and these two green is the cis. Okay, so this is a cis form. Okay, but if it is in the trans form, so whereby the chlorine and amino, okay, switch each other, there is no anti-cancer activity at all. So you just imagine, okay, with this very tiny, tiny, tiny little molecule, just by switching the position, one work as an anti-cancer drug, which you can sell it, you know, with a very high price. But another one is, I would say that is a useless, okay, totally useless. Cool. So that's made me impressed with this, you know, the chemistry of the drug. Okay? So from with that interest, I further study, as I explained, to master in the PhD level, and that is where I mastering, you know, many synthesis techniques and also analytical techniques. Okay, and it's so blessing, okay, with this career, okay, that I have opportunity to work with culturally diverse teams of researchers and scientists. Okay, and one of the best moment is when I spent my 6-month attachment in IBM Almaden Research Center in Silicon Valley, California, and able to come up with the one patent from that attachment, which I will share later in this talk. Okay. So now come to a little bit more about the story of this cisplatin. Okay, so on the screen now you can see the example of Food Drug Administration America that approved organic metallic complex in cancer therapy. Okay, so on the left top, again you see the structure of the cisplatin, and at the bottom there is the, you know, the picture of the cisplatin as how it is available clinically. So if you go to the pharmacy or if you go to the hospital, okay, you as a layman or normal person, whenever you see drug, you will only see the drug in the form of the you know, the physical form. Okay, like cisplatin one in this picture is in the form of liquid. Okay, so that's how laymen look at the drug. Okay, they only can see it in the form of physical form, like this one in the liquid. You can see in the form of tablet, you can see in the form of a capsule, for example, or in the form of the cream. But we pharmacists, okay, who basically we learn about the chemistry of the drug, we always look at the drug, okay, in the form of the structure. Okay, so this is the structure of the cisplatin. You can see the mechanism of the action, okay, as available on the screen now at the center. So how this cisplatin, okay, if you can see my pointer here. So the cisplatin, after it went through or passed through the membrane, okay, the cell membrane, okay, it will went through a few modification, okay, and then after went through this few modification, it will go to the targeted DNA. So in DNA, if you learn, okay, there are a few bases in DNA. So which bases that this platinum drug will targeting is the G base. So as you can see that how the platinum here make binding with the G base on this DNA double helix. Okay, and there are few modes of binding, like how you can see on the screen now. So by make binding to the DNA stranded of a cancer cell, this will disrupt, okay, the further process that related to the DNA. Okay, so like DNA replication, etc., etc. So that will eventually kill the cancer cell. Okay, and because of the cisplatin, even though it is considered as a super drug, but because of a few side effects. So scientists or medicinal chemists like us, we keep exploring and exploring in further improving this drug. Okay, so that is where the other drugs that related to the platinum come up, such as carboplatin. You can see here, carboplatin, and also oxaliplatin. And if you notice that the basic structure is still remain, okay, you can still see there are two amino group, okay, bind to this platinum as well as here. Okay, So this is how the modification took place. Okay. All right, in this slide, I'm going to share further a little bit, but in the very basic term about how the drug discovery and drug development process take place. Okay, so you can see from the left side here that first of all, we must find the target selection of the drug. Simply said, okay, like the case of the cisplatin just now, we know that the target selection is a DNA double helix. All right, so that's require many information. Okay, like the genomic information or genetic information. So these are a few targets selection that are available for the drug. It may target the cellular or genetic, like cisplatin, also genomic, also proteomic, okay, some protein, and some of these target also can be what I call this can be studied or targeted by from a bioinformatics information. Okay. And then once the target selection been identified, okay, medicinal chemists, okay, we start create or synthesis small molecule. Okay, if so, what we do is the synthesis and then do the isolation, and we also can use with the combinatorial chemistry method, okay, and the assay development method to determine a lead compound, okay, or lead discovery compound. So, and from here, it is developed into library development, okay, of many compound, and then from there, we conduct what we call a structure activity studies. So, like just now, okay, in the cis position structure, we know that it works, and if it is in the trans position structure, we know that it won't work. Okay. And then also there is in silico screening process and chemical synthesis process. Chemical synthesis process in the way of finding the most optimum pathway, okay, in synthesize a new drug. Okay, we don't want to spend so much or so long route to create a new drug. Okay, we want to shorten the process. Okay, we want to find the optimum what I call this a chemical reagent. Okay, so that is where the medicinal chemist skills take place. Okay, and then this drug discovery process further into the in vitro studies. Okay, in vitro studies is to determine the drug affinity and the selectivity, also to determine the cellular disease model, okay, and mechanism of action, so on and so forth. Okay, and this will further lead to the candidate refinement. Okay, and then after certain data, okay, certain what I call this promising data obtained from in vitro study, it will be elevated into in vivo studies. Okay, so these in vivo studies will involve, okay, live thing. Okay, so animals, okay, animals models of disease stated will be used here, okay, in these in vivo studies. And then in this stage, okay, there are many information that we need to obtain or get from these studies, okay, such as behavioral studies, etc., etc. And after we confirm, okay, the effectiveness in these two stages, in vitro and in vivo, then the candidate will move into the clinical trials and therapeutic. So clinical trial is, as I think everyone know about the top issue or top news now with regard to the vaccine. So how this vaccine, okay, required a few stages of the clinical trial. Okay. All right. Here is I would like to talk a little bit about what is a lead compound that we've been talking here. So lead compound is a compound that having a particular biological activity obtained either from natural or synthetic source. Okay. So the example giving here is penicillin G. So this is the structure of the penicillin G. Okay. I hope you don't feel that difficult to look at this structure. So penicillin G, known as a lead compound, which is obtained from natural source. It is not an ideal drug because of the acid unstable. Okay, it also showing narrow microbial spectrum with high allergenicity. Okay, and also develop some of the drug resistant. So due to this problem, penicillin G is not used here. Okay, so what happened? We medicinal chemists, okay, we do a modification, okay, on this structure of the penicillin G. So what we did is we add another group, okay. So one amino group, okay, been added here. So by adding this one simple so-called functional group, okay, this penicillin G, okay, turn into ampicillin. So ampicillin is a result of molecular modification of penicillin G and reduce many problems with penicillin G. Okay? And so with all this improvement, now ampicillin being used instead of the penicillin G. So this is how we medicinal chemists work. Okay? And finding a lead compound, okay, there are many ways, okay, in finding a lead compounds. I give a few examples here on this slide, like a natural product. Okay, there are also lead compound that we can find by screening of natural product. Example given here, Taxol, okay, is a little bit complex structure, but do you know that Taxol actually discovered from a big tree? It's a very big tree. It's a skin of the tree, okay, that having this compound called Taxol. And can you imagine that? Do we have to, cut all these tree that having Taxol in this? In order to produce more Taxol drug? No. Okay, so that is where we medicinal chemists, okay, will do the synthesis of this Taxol. Okay, with this complex structure, we will do at the lab, try to synthesize this Taxol. And a matter of fact, okay, after this session, you can try to Google synthesis of Taxol, and you may find there are many ways of synthesis of Taxol. Okay, that's happening in many laboratory now. Okay, so the aim is to find the most optimum synthesis route, okay, to produce this Taxol. So that is one example how this lead compound derived from the natural product. Other examples such as a synthetic compound, okay, and then also chemical modifications of natural ligand. Okay, so I give example here for chemical modification of natural ligands as adrenaline. So adrenaline is actually a natural neurotransmitter in our own body. Okay, so there are many drugs that derive from this adrenaline where there are some modifications happen on the adrenaline structure, so that this new compound or new structure will act as, for example, adrenergic beta agonists, okay, such as salbutamol and dobutamine. Okay, so if you can Google salbutamol structure, you may find there is a similarity with the structure of adrenaline. Okay, so this is how the chemical modification of natural ligand took place. Okay, and sharing about the recent metal complex in cancer therapy. So this is one literature that I pick up, okay, published in 2019, okay, from one group in a very well known journal. Okay, so here it is showing, okay, the a little bit complex structure. Okay, you can see the big star here, named as HPPB. Okay, and the illustration here, and you can see the DSTP and also the CPT. So this CPT, if you notice that it has a platinum, okay, the platinum metal ion at the center here. Okay, and there are a few other molecule components. So the combination of all these components actually will self-assemble together, okay, like what you can see on the screen here, the M. So this M structure actually providing a space. So this space, okay, will be filled up with this OEP. So this OEP will be filled up in this structure M. So as you can see on the screen here, and then with further combination, okay, by using a few polymer like this and assemble together, it become what we call a magnetic nanoparticles. Okay, so this magnetic nanoparticle, okay, when injected into the mice here, okay, as a model, when it's traveling into the bloodstream, okay, and then enter one particular targeted cell, okay, like this, and when this system being exposed with the laser light, okay, so it release, okay, everything inside the cell, and this is where, okay, you can see that the platinum, okay, that is the platinum so-called drug here take action. Okay, so this is how the latest or recent mental complex in the cancer therapy study. So with this, we can find way in how to target a particular cell, okay, that related cell, so that it won't go to the other cell, the other healthy cell, etc. Okay. I think this is my last slide, okay, that I would like to share about my recent achievement in a co-formulation polymer drug nutraceutical. So as I share in the earlier part of this talk, that I spent 6 months in IBM Almaden Research Center. So this is, you can see the IBM logo behind me here, okay, with and then this is the photo, the actual photo. I spent my time in the laboratory there. So this patent was registered in the United States on the title Co-Delivery of Cholesterol-Lowering Drugs and Nutraceutical. And you can see the applicants here, International Business Machines Corporation, IBM, okay, and Nano Malaysia Berhad, the company that sponsored my research attachment at that time. So you can see the inventor's name here, okay, my name as a first inventor, Mohd Zulkefeli, and then this work was performed with other actually multinational scientists that work in IBM, Dr. Els Hakim Nelson, Dr. Victoria Punova, and Dr. Joseph Sly. Okay, so the study was about one particular biodegradable polymer that invented by IBM. So my work at that time was to do the co-formulation, how to make this one drug, okay, the um, cholesterol lowering drug, simvastatin, and also one nutraceutical that related to the cholesterol as well, which is ubiquinol, or known to public as a CoQ10. Okay, I think if you go to the pharmacy, you can find this CoQ10 available as a supplement. So my work at that time was to formulate these two, okay, drug and nutraceutical in this one polymer, so that this polymer will work as a carrier. Okay, and this work was able to register as a patent in United States. Okay, I think that's all that I wanted to share in this session. I pass the next to Dr. Pecina.
Right. Thank you, Prof. Zo. Before we move to the Q&A section, I wish to share, right, some information regarding the Bachelor Science Pharmaceutical Chemistry program, all right, that available in IMU. And indeed, this program is actually a ticket, all right, which will lead you, to embark to the journey of drug discovery and development, all right. So IMU Pharmaceutical Chemistry program is actually the first undergraduate 3-year degree program in pharmaceutical science that's being locally offered in Malaysia. Okay, and then this program is indeed aimed to provide interdisciplinary field of study and also training for chemistry undergraduate students. Okay, so the student in this program are exposed to different areas of chemistry, especially synthetic organic chemistry. All right, and not only chemistry, actually the students will be taught regarding pharmaceutics, okay, which explain about different dosage, like, you know, cream, why some drugs has been formulated into cream, why some in tablets, some in capsules, etc. All right, and the students will also be taught regarding the pharmacology aspect as well as other biological specialties relate to the drug formulations and drug delivery. Okay, so from there, all right, the student will then understand the different chemical structure of medicines and how the molecules that, you know, react with each another. So in a nutshell, okay, the students equipped with knowledge and skills in design, chemical synthesis, analysis, and, you know, formulations, you know, for market of pharmaceutical agents or bioactive drugs. All right. So pharmaceutical chemists or the graduates, all right, graduated from this program will plays a significant role behind the discovery, development, as well as the formulation of medicines. All right. Okay. For the career pathways, okay, so graduate from this BSc Honors Pharmaceutical Chemistry program can work in different fields, okay, be it in a position in laboratory or non-laboratory setting. So the pharmaceutical chemistry program prepares their graduates to contribute to the research and development in the pharmaceutical industry, all right, as well as you know, their current primary roles in quality assurance, quality controls in pharmaceuticals. All right, so the PC graduates can actually also work in laboratory affair, marketing department, or even in academic. All right, so as the program also equips the student with the fundamental knowledge and skills in chemistry, it actually also enables the graduates to work in other well-developed or high potential industry in the country. So these include cosmetics and pharmaceutical cares, or even polymer industries. All right. So in the subsequent slides, all right, so I would like to share a few of pharmaceutical chemistry alumni who involved particularly in the pharmaceutical chemistry or pharmaceutical research. So the first alumni that I would like to introduce to you is Eric Yong. So Eric graduated from pharmaceutical chemistry program last year, all right? And how he involved into this pharmaceutical research is through his internship, you know, training at the Oncogen Pharma, which is a research-based pharmaceutical company with a global outlook, you know, to prepare the cancer treatment drug. And because of his outstanding performance, all right, he was actually offered, you know, a permanent position as a formulation executive in the formulation development department in Oncogen Pharma. The next alumni that I would like to share to you is Zen Way. So Zen Way also embarked into, you know, his career in research and development, all right, through his internship experience in Cancer Research Malaysia, all right, which is the cancer research organization in Malaysia. So his project is, of course, related to the cancer drugs. And then with the interest that he developed, all right, from the internship, subsequently now he is, you know, pursuing to higher degree and is doing his PhD now. So another graduate from IMU Pharmaceutical Chemistry program is Kiki, all right. Kiki completed her pharmaceutical chemistry degree, all right, as well as a master in medical and health science from IMU. And then Kiki shown a very strong interest in medicinal chemistry. And she has published more than 28 research papers, throughout her degree as well as master studies. And now she's one of the lecturers in the School of Pharmacy. All right. So let me share to you another uniqueness of this particular program. Okay. So IMU Pharmaceutical Chemistry program has very close relationship with ex-partner universities. All right. So we have the partner university from Australia, namely Curtin University as well as the Sydney University. So the graduate from the BSc Pharmaceutical Chemistry, all right, so they can actually pursue their study, all right, once they completed their 3-year undergraduate program, then they can continue to pursue their study in Master of Pharmacy, all right, from these 2 universities in Australia, the Curtin University or the University of Sydney, and then graduate as a pharmacist. And the graduates from these 2 universities, then they are registrable for practice as a pharmacist in Australia, New Zealand, as well as Singapore. And last year, all right, we have actually developed a new partnership with the University of Dundee from United Kingdom, okay, UK. And then in this particular track, all right, so the students is actually allowed to transfer their credit, which they learn from the first year of IMU Pharmaceutical Chemistry program, and then into the 2nd and 3rd year of the sciences programs in the University of Dundee. And this entire, partnership actually prove that the quality of the program, that is recognized nationally as well as internationally, because it's not only, able to do credit transfer or credit recognition into those universities, but IMU Pharmaceutical Chemistry program is also accredited by Royal Society of Chemistry from the UK, as well as recognized by the Institute Kimia Malaysia, all right, locally, okay. So, well, so subsequently is Annette, all right, we are proud to introduce you Annette. Annette is also, she's an international student from Uganda. She completed her Bachelor Science of Pharmaceutical Chemistry degree in IMU, and subsequently she continued, her study in MPharm, all right, and then with credit exemption, of course, all right, and then she has completed, all right, his MPharm in Curtin University, and now she practiced in Australia, and then she stayed in Australia, all right. So the other example is like Wayne, all right, as well as Simon, okay. Both of them also completed their degree, their 3-year pharmaceutical chemistry program in Malaysia, all right, and then they decided to pursue their study and then to the MPharm degree in University of Sydney, and then now they have graduated and also they continue, all right, to work or practice as pharmacists in Australia. Okay, so well, I think those are the examples that I would like to share to you, and some informations that I would like to share to you also regarding the IMU Pharmaceutical Chemistry program. Perhaps we start with the first question, Prof. Zoh. Well, I think this is an interesting question. Okay, so in your earlier presentation, all right, Prof. Zoh, you mentioned that you love biology so much, right? and then and you indeed you you call yourself a biology nerd. And I wonder actually, what really changed your mind that you switch from biology minded to chemistry minded? Because for me, I have opportunity to meet many students, even though the high school students, and to them, wow, chemistry is nightmare. All right. So, but in your case, perhaps Dr. Zhu, you would like to share to us like how you change your focus right from biology to chemistry. Yes, Dr. Zhu, please.
Okay, thank you so much, Dr. Kim Pecin. Okay, that's matter of fact. I always receive that question in my many talks when I share about my experience. All right. Um, well, actually, as we know that in this pharmaceutical science program, okay, we learn actually both biology-based module as well as a chemistry-based module. Okay, so we also learn like pharmacology, right? And then we also learn like microbiology. Okay, understanding the cells, understanding some of the organ, okay, the physiology of the organ, etc. And then when come to the experiment, okay, many of these biology related experiment will be using animals, okay, like mice, rat, okay, and even like a guinea pig. Okay? And in my program, when I studied in Japan, they even at one time use a dog. Okay, so I will consider, okay, because of my background as a biology nerd, I can consider that I am pretty good in handling animal experiment. Okay, but somehow I just don't feel comfortable. Okay, um, so I think because of this feeling, okay, a general feeling that I don't feel comfortable, you know, doing the animal experiment. And when I went into many of these chemistry-based module experiment, okay, like doing synthesis or analysis, okay, there is basically no live thing, and I somehow feel enjoy and probably a little bit obsessed statement that when I do synthesis and when I study about the molecule, I would say that I actually talk to the molecule. So like when I do the synthesis, okay, from reagent A with reagent B, okay, to produce compound C, for example. So while performing this chemical synthesis, I like talking to the molecule, okay, like, okay, please make a combination together and then produce this compound C. So I think that's what made me don't feel um, fear, okay, about chemistry. Okay, and some more, when you learn chemistry in the form of the structure at the beginning, is actually will make you feel easy, or make you feel that you are closer to chemistry, instead of you learn it by the word itself. Okay, so I always telling my student, okay, if you, for example, if you learn about methanol, don't learn by the word methanol, but learn about the CH3OH structure. Okay, and then learn that the three H atom connected to carbon, and then you have another bond which is oxygen, and then this oxygen connected to the last hydrogen atom. So if you make yourself familiar with this structure of methanol, okay, you won't feel fear when you look at the structure of the methanol or any other structure of compound or drug that you may learn. So I think that's helped me a lot in um, changing from biology to chemistry. And another thing that I feel that there is true in this is what my supervisor told me, that if you further study in the chemistry line, you can always back to the biology. You can always learn and understand the biology. But if you further into the biology area or biology field, it will be difficult for you to turn back to chemistry. So it's actually become a chemist, of like myself now, medicinal chemist. It is advantage because let's say when I want to study about the target of the drug, which is uh, basically biology molecule, okay, the cell, the DNA double helix structure. So I can able to study back about the biology of those. Okay, but I think it is difficult for a biologist to you turn back to study about the chemistry. So I hope that my experience will be useful to the students, okay, to decide about your pathway.
Yes, certainly. Thank you, Prof. For sharing about your interest in both biology and also in chemistry. And I learned something, that is to make friends with your with chemistry, right? I think that will make your life more interesting, okay? And then it's easier, all right? So, and actually, indeed, my supervisor, when I did my PhD, also shared to me like the wider career prospect with the chemistry knowledge that you have, because when you zoom down into different areas such as biology, so in the body system, it's also because of the chemistry, right? Make the reactions that happen. Yeah. Thank you, Professor, for
Yeah,
that sharing. We move on to the second question, the drug discovery process. Um, actually, looking back at this drug discovery process, right, I was wondering, Prof. Su can elaborate a little bit more what a pharmaceutical chemist, right, can do, or perhaps what is the role that they can play in each of these drug discovery process.
okay, thank you, Dr. Pecina. Thank you for that question. I think I would like to begin, okay, with the most left about the target selection. So as a medicinal chemist, okay, um, what we can do is once we know what will be the target of the drug or the disease, okay, then we can start study about what molecule that having interaction, okay, with this target. So, for example, taking example of DNA double helix, okay, in the case of the cisplatin. So, because of the size of the structure of this DNA double helix, it is commonly, okay, the drug that will be designed is in the group of what we call a small molecule, okay, because of the small molecule. They're able to reach the DNA double helix. Okay? But when it comes to, for example, protein, okay, so protein, okay, as we know that, for example, enzyme uh, is also under the class of protein, and uh, enzyme, how the enzyme works, okay, usually enzyme will have like what we call active site, okay, inside it. So as a medicinal chemist, we also can, maybe using like computational chemistry method to identify what kind of small molecule that can fit in, okay, into this pocket, okay, so that when this molecule fill up this active pocket, okay, in this one particular enzyme, the activity of the enzyme may be stopped. Okay, so that is in the form of identifying, okay, the what will be the right molecule that fit with the target selection. Okay, and then next to the lead discovery, the role as a medicinal chemist, okay, obviously is the skill in the synthesis. Okay, so synthesis is in the way of, like in the case of the Taxol, is to find the optimum what I call the synthesis pathway. Okay, because of in order to synthesize such as a complex structure, it requires many steps. So as a medicinal chemist, we work on to minimize the step. For example, okay, if it requires 15 steps, so we try to minimize, okay, to make it shorter, from 15 to maybe 10 steps, or from 10 steps to 5 steps, for example. And then because that will save lots of costs, okay, that required in the synthesis process. So that is in the case of the Taxol, okay, where the exact structure is already known. For example, combinatorial chemistry. So medicinal chemists can do like combine with so many candidates of compound, and then do the what we call it like cross checking. Okay, so when we do the cross checking um, synthesis, for example, like one particular compound, we attach with many types of what we call it functional group. Okay, so like the main structure with functional group A, the second main structure with the functional group B, then continue with the C, D, E, F, okay, something like that. So with this combinatorial chemistry, okay, is also another way in find the lead discovery. So that is also what the medicinal chemist can do. Okay, and obviously for the next medicinal chemistry part, okay, again the chemical synthesis, in silico screening, okay, in silico screening. So you need to learn something that related to the computer, okay, and then structure activity studies. There are many steps in the drug development or drug discovery process that require medicinal chemists to do many testing, okay, and even in the in vitro studies, okay, in determine the mechanism of action. Medicinal chemists, we work together with the pharmacologists. Okay, we work with the pharmacologists, and when come to the in vivo study, also we work together with the pharmacologists, and when come to the clinical stage, okay, this is where medicinal chemists will get further information about the effectiveness of the drug that went through the clinical trial, okay, to see how medicinal chemists can, if required, need to do modifications on the structure, etc., etc. Okay, so I hope that's answer the question, Dr. Kempisi.
Yes, of course. Thank you, Prof. Zul. From IMU Farm Camp, there are also a few graduates, right, from this program that actually involve or work as the clinical trial associate when they graduated from pharmaceutical chemistry program. I think one thing that I would love to have from Prof is actually motivation or encouragement, especially Prof, for those youngsters, all right, in this talk, that whether they have some or whether they have great interest in research and development, right? So what can you share with them? For example, your encouragement or your tips of, of secret of success in this, research and development journey. All right. So if you can share to them, Prof.
Okay, thank you, Dr. Pecina. To me, I think what required is you need to make a first move or first step. Okay, so in my case, okay, my first step is, of course, I entered the pharmaceutical sciences program. Okay, and then from that program, I actually learn, okay, all ranges of this pharmaceutical sciences area. So that is where I able to identify where is my interest. Okay, So as I share many times that even though I was a biology nerd, but I found that my interest, my real interest and ability is in the chemistry. Okay, so then from there, I was lucky. Okay, that I able to find the right research laboratory at that time. Okay, and then from there, I just move ahead. Okay, I just look forward. Started from equip myself with the chemical synthesis skills. Okay, and then from there started with one compound, goes to the next compound, third, and then similar to the publication as well. And matter of fact, if I may share here that my publication that come up from my PhD project, that was in 2005, so it's already 16 years back. Still, I get a citation of that publication even until now, and it's counted more than 100 citation already. So from one thing that you discovered at that time, people are still referring to your discovery, okay, for their works. So I think that is the beauty of involved in this research area. So I would like to encourage youngster, make your first move, which is obviously come to this pharmaceutical chemistry program. Okay, and then from this pharmaceutical chemistry program, okay, after you learn many things, then your next step, okay, will be, you will be able to determine your next step from there. Okay, that's it. Thank you.
All right. Thank you, Prof. Zul. I think it's the self-satisfaction as well as the contribution to the well-being of the society that actually keep the scientists moving on. Right. I think it's now come to the end of the live talk, Prof. Right. So thank you very much, Prof. Zul, for such enlightening talk. And then thank you everyone for participating in this live talk. Thank you all for your attention, and then have a lovely afternoon. Bye-bye.
Thank you so much. Thank you.